Okay, hi, I'm Ben Freeman. I'm teaching the class BIO418 while Dolph is out of town. I had some technical difficulties recording the class yesterday, and so I'm going to record this little Zoom version of the material that we covered in class yesterday. I'm going to go pretty quickly, so if you're watching this at home, I'm going to tell you at different points to pause, think about something. If you want to learn the material, please do so. So today, the goal of today is for you to understand the mutation accumulation and antagonistic pleiotropy hypotheses to explain the evolution of senescence, know how they're similar, how they're different, what they're all about. And so the topic is the evolution of senescence, which is the physiological deterioration that accompanies aging, and it leads to accelerated mortality and decline in reproductive performance. The question is, why does senescence occur? And you might think that, well, it's just what happens. It's unavoidable, inevitable consequence of wear and tear. But we actually have lots of evidence that senescence is a thing that evolves. These mutation accumulation and antagonistic pleiotropy hypotheses, the basic idea underlying them is that the strength of selection declines with age. And so that can lead to the evolution of senescence. And some evidence that senescence is not a fundamental constraint comes from hydra, these little freshwater animals that live throughout much of the world. There's a record of them in iNaturalist from Pacific Spirit Park nearby. And they're apparently immortal. So what this graph is showing you for different species are Drosophila. So at age 0, on the y-axis is mortality. The Drosophila are all still alive. None of them have died. And then it goes up. And by about 100 days, Drosophila have died. Drosophila do not live 100 days. Microtus is a vole, a little rodent. And they started off being all alive in this lab population. And by maybe a year or so, all the Microtus have died. All the voles are dead. And that is the common pattern in these lab organisms. Hydra, on the other hand, have just this flat line. They're all still alive. And they did this multiple times for different groups, different controls. They're all still alive. And the interesting thing about Hydra is that they can reproduce sexually. They usually reproduce asexually by budding. And the interesting thing about Hydra, really, on top of that, is that they almost any cell in their body, I guess the idea is any cell, can regenerate a new individual. So they don't have a clear separation between germline and soma. They also show no obvious signs of intrinsic senescence. That's what these graphs are showing you. So you might think it's the distinction between sexual and asexual reproduction that's really key here. But the authors argue it's really the distinction between the soma and the germline. That in the Hydra, there is no distinction. All cells are germline. But once you get that distinction between soma and germline, the idea is that evolution of somatic differentiation can create the conditions then when the soma can senesce. And that's really what we mean when we talk about the evolution of senescence. And so that's the disposable soma. Okay, so we have evidence that it's not a fundamental constraint, and we have lots of other evidence beyond Hydra that different species vary in their rates of senescence, suggesting it's something that evolves. So the two hypotheses to explain it were both first articulated by Peter Medawar, totally fascinating guy, Lebanese British, born in Brazil. Check out his Wikipedia page. He was an immunologist. He won the Nobel Prize for his research on organ transplants. Been termed the wittiest of all scientific writers. One of his epic takedown burns from the 1930s said, in a book review, he said, its author can be excused of dishonesty only on the grounds that before deceiving others, he has taken great pains to deceive himself. He got his PhD. I mean, he got a Nobel Prize, right? So he got his PhD, he did well. He never actually received it because it cost $25 to get the physical plaque or whatever it was from his university. And he didn't have any money at the time because he had just zeroed his finances paying for an emergency appendectomy. So his first idea is this mutation accumulation hypothesis. And he explained this by doing a thought experiment about test tubes. So, he worked in a lab, there's these test tubes. So imagine test tubes break. Sometimes they get dropped, shatter, break. So they break at some rate. And this breakage rate is unrelated to how old the test tube is. And so in this scenario, you're going to get an age distribution of test tubes that looks something like this. You have a lot of test tubes that are young and some test tubes that are old, that have been used for many months. So the experiment, the thought experiment goes like this. Now let's assign some sort of fecundity to test tubes so that they have a stable, test tube population. Let's make them little replicators. This fecundity is unrelated to test tube age, and we can even imagine giving these test tubes genes. And consider a mutant test tube gene called brittle that makes it more likely to break. Okay, so this mutant will be at a selective disadvantage. Now consider a second gene that alters the timing of expression of brittle. And the second gene comes in two alleles. Mutation A causes brittle to be expressed at an early age, and mutation B causes brittle to be expressed at a later age. So what I want you now to do is think about this question: which mutation will experience stronger selection against it, mutation A or mutation B? And explain why. So pause and answer that, and then resume. Okay, the answer is mutation B will be selected against at a lower rate than mutation A, simply because fewer individuals, fewer test tube individuals live long enough for this mutation B to impact their fitness. And so the result of this is a disproportionate accumulation of deleterious mutations that are expressed and act on older individuals. In this thought experiment, older test tubes. This idea that old age is a genetic dustbin. And here, mutation accumulation refers to accumulation over evolutionary time, not an individual lifespan. So again, the idea is that these mutations that have negative effects that are only expressed later in life will experience relatively weak selection against them. They'll still be selected against, but they may be able to persist in populations at mutation selection balance at appreciable frequencies. Okay, so mutation accumulation has said that here's a gene that doesn't impact young individuals and has a negative impact on older individuals, and thought about the conditions in which that gene might persist in a population. Selection will be against it, but won't be able to drive it to zero. So antagonistic pleiotropy. I want you to take a minute and think about what you think antagonistic pleiotropy is going to be. So pause and think about that. So antagonistic pleiotropy is a gene that has a positive effect on fitness early in life and a negative effect, a deleterious effect later in life. So pleiotropy means one gene that has multiple effects. In this case, they're antagonistic, positive early, negative later. And that same declining force of selection with advancing age that I just talked about with the test tube analogy means that this gene here that has a positive effect early on and a negative effect later on means that gene might actually experience positive selection. The soma becomes disposable. If it has a positive effect early on, and a negative effect later, that might be enough for that gene to actually be favored by selection. Okay, so those are the two big ideas that Medawar proposed. And so both of these imply then that the evolutionary pressure that leads to senescence will be highest when the overall rate of mortality is high. This idea could explain variation that we see among species in rates of senescence. There's some details of theory people are still arguing about details, but some assumptions like population growth must be density dependent seem to be generally met. Important here, there's no genes that are like for aging. These are ways in which genes that cause aging can be maintained in populations, either at mutation selection balance or actually be favored by selection. And so we can then think about this relationship between rates of mortality and rates of senescence by multiple routes. And so the first route is by comparative study. And so this study looked at there's these percent surviving curves for a bunch of different birds. They actually did a bunch of different mammals as well. And from this data, first of all, it shows that different species differ in their rates at which they senesce. And from this, it can calculate rates of senescence and mortality rates. And so then each species can be plotted on a scatter plot of rate of senescence on the y-axis versus baseline mortality rate on the x. And I want you here to think through what's the predicted relationship and why. Explain your reasoning. So pause here, answer this question, explain your reasoning. Okay, the answer is that it was a positive relationship. So those, again, each species is a point, and those species with higher baseline mortality rates also have faster rates of senescence, suggesting that support for this idea that mortality rate is an important driver of the evolution of senescence. Oh dear. I didn't mean to bring this one up, so let's wipe that out. Okay, so that was some evidence about the importance of mortality rates from a comparative study. This can also be addressed in selection experiments. So for example, this particular selection experiment was on Drosophila, and they did artificial selection. They took a CO line, O you can think of for old, where they only took young from old Drosophila. Old Drosophila are more than 3 weeks old, over 21 days. They also took Drosophila from the young line, B. You can think of B, CB, B for baby. They took these from these young fruit flies that are eggs produced when the adults were 1 to 6 days old. And right now I want you to pause and think through, what do you predict the consequences will be for this, for the evolution of senescence? Well, it turns out that this had consequences. The CO line evolved longer lifespans of 30 versus 26 days, and moreover, this had strong consequences for life history traits in these fruit flies. So this plot shows time on the x-axis, how old the fruit flies are, and on the y-axis, it's the number of eggs of the CO, the old line, minus the CB, the baby line, of these two selected lines for each day. So if the old line at a particular age lays more eggs than the B line, it'll be a positive value. And that's what we see for these older ages. So these fruit flies that are 15 to 25 days old, the ones in the CO line lay more eggs than the ones in the CB line. And so I want you to pause and come up with your prediction of what we're going to see on these younger ages, say ages 0 to 15. Well, here's what they found. There's actually a flip. So the ones that were the CB line, after selection, they lay, not only do they not live as long, but they lay way more eggs at young ages. So this is pretty fascinating, these kind of far-reaching life history consequences of being selected for different mortality, and it tells us some fascinating things about the evolution of senescence. To me, it kind of suggests antagonistic pleiotropy, that there's some genes that are better for the young flies, and some that are better for that might have negative consequences for the older ones, and those are maybe selected out in the CO line. But there's other interpretations of this data as well. And these sort of patterns have been widely found in Drosophila. So lifespans increase when you select on time of reproduction. So this is kind of, just showing you that there's these interesting relationships between the time of reproduction and lifespan and when reproduction is occurring within these species. So now let's move to trying to test some of these predictions of the hypotheses. Okay, so I've shown you how senescence can evolve. It's related to mortality rates, both in comparative studies and when humans have kind of imposed mortality by only taking eggs from certain ages. It's as if in those baby line that the mortality was at 6 days, and it's if in the old line that mortality was only for the older age. Okay. So now let's see if we can test some predictions. And here's a paper that went through a mathematical model and came up with some predictions. They said under mutation accumulation, inbreeding decline, and 3 major components of genetic variance are expected to increase with age in randomly mating populations. And so there's a test of this with these snails. And so the idea is that inbreeding, what it does really is inbreeding exposes recessive deleterious mutations. And so the study tested the effects of inbreeding on this snail that's a hermaphrodite. And so the argument is that these deleterious effects of inbreeding, quantified as inbreeding depression, should increase with the frequency of expressed deleterious mutations. And in the mutation accumulation idea, these expressed deleterious mutations, there's more of them at older ages than younger ages. And so we might expect to see the effects of inbreeding depression greater at greater ages. And that's exactly what they found in these snails. And they went further and looked at heterosis. Okay, so heterosis is heterozygote advantage. It's, again, thinking about these recessive deleterious mutations. But here it's pointing out that if in a mutation accumulation world, different populations will accumulate these deleterious recessive mutations that are expressed later in life. But then if you cross one population with another, the resulting progeny will have each of those deleterious recessive acting late in life alleles from each of their parent populations masked by the wild type allele from the other. So instead of having two copies of the recessive, they'll just have one copy of all these recessives, and those won't actually be expressed. And so there's greater numbers than of deleterious mutations should lead to greater heterosis when hybrids are made between populations having different deleterious mutations. So under this mutation accumulation hypothesis idea, the effects of heterosis should increase with age. And that's exactly what they found in these snails. So some positive evidence for the mutation accumulation hypothesis. And I just want to pause here and say that this is a really active area of inquiry. There's lots of folks trying to study the degree to which mutation accumulation or antagonistic pleiotropy or the relative balance of these two ideas in explaining the evolution of senescence. There's vigorous debate continues on the relative importance of these ideas for explaining the evolution of senescence. But more recently, we can do a little bit better than just have thought experiments about test tubes and say, imagine a gene that does this. We can actually kind of look at some of the genetics that underlie the evolution of senescence. And I'm going to close by showing you some of this data. Some of this is phenomenal to me. So what this data is showing you, this is for some nematodes, some C. elegans in the lab, and the wild type is shown in black. So the wild type here is percent alive on the y-axis, so they're all alive at day 0, and then they die. So they senesce and die pretty quickly by 20 days. But if they have a DAF2 mutant in the top panel, they live over twice as long. And if they have a DAF2 and a CLIC1 double mutant, they can live to 80 days on occasion. So two mutant genes, and they can live 3-4 times as long. Totally amazing. And the bottom panel, B, from the same study, looked at another modifier gene that modifies the evolution of senescence called ISP1. And the point here is showing that ISP1, when you have ISP1, these nematodes live longer, but it doesn't have a positive effect in conjunction with DAF2. So the double mutant DAF2 ISP1 has just the same curve as just DAF2 alone, whereas the DAF2 CLIC1 gets an added benefit beyond that of DAF2. So illustrating some of the epistatic interactive effects between genes that modify rates of senescence in these nematodes. So this sort of work tells us that, at least on occasion, we might have single large effect genes that impact the evolution of senescence. Okay, so that concludes my recap of the material for BIO418 about the evolution of senescence. If you have questions, you can contact me on Slack or via email, and I'll see you in class next week. Have a great Remembrance Day, fall break, and weekend. Thank you.